Okay, questions? Anyone? Yes, sir.
On the exam and quiz last week, there was a question about the growing techniques, something about paraphrasing the growing in solution, and I think one line of the most popular technique.
Uh-huh.
Was that hydroponics or was that?
I'll go over that a little bit later. I still got one person that's going to take the quiz or take the exam. So, but I'll go over that, and then you should get. You also once it's past the due date, which you can go into your grade and see your exam, and then you can see, and then you can come and ask me. You can either email me, and I can change the answer, or you can come and see me. Okay. So you just go into I know it's like everything else's system, it's a little odd, but you go in and you can see your scores, what you chose, and the correct answer. And I assume, I can see mine, so I assume you can, I'm demo student, so you guys can see that, right? You can see, okay. Yes, ma'am.
When is the take-home portion of the exam going to be?
Wednesday or Friday at noon. I sent it out in an email. Was it Friday at noon or Friday before class? Was it Friday by class time? Yeah, I sent an email out. Okay, and I haven't put the column up yet. So the take-home portion Friday before by class time. Okay.
Do you know when you're going to do that?
Probably sometime later today or this evening. It'll just be, again, work your answers out and then copy and paste. And if you have, and a couple of people have had problems where the computer locks up or whatever, if you let me know, I can go in there and reset for you to do it again. And there's actually some things that it goes in and says computer, I don't know how it knows, but it says, it's not like its test was terminated by something. One person had their computer just die in the middle. And a couple of people have had Windows update in the middle of when you're taking the exam, and actually it's and it's crazy. Windows updates Wednesdays, I think, unless there's a critical update. So if you happen to be, if you don't have, if you have it set the way it should be, which is to automatically stall the updates, then, and that's actually happened a couple of times. I know that's a weird problem, but, you can get around that, but it's really not worth it. Okay. Any other questions? Okay. So let me backtrack here a little bit and put this stuff together before we start talking about actual translocation. And this is back from the light reactions, and the point to emphasize here is that this occurs in all plants. This is C3 photosynthesis, the carboxylation, the reduction, and then regeneration. So when we talk about C4 and CAM plants, they have this second set of carboxylation reactions that occur. Now, I wanted to go and show you this also. This is from the light reactions lecture, and it's a good comparison of the C3 and C4. And I think when I went over this, I said we'd be talking about it again. So if you look at the C3 here in the palisade cells, this is where the photosynthesis occurs. You can see they're fairly dark because of the chloroplast. And then a little bit of photosynthesis will occur in the mesophyll cells, but mostly in the palisade cells. If we look now at a cross-section of a C4, and again, it's sort of the darkness here, and you can see here, is now you have these chloroplasts that are sort of scattered throughout the leaf where the first carboxylation reaction occurs. The C4 organic acid is produced. That is not inhibited by oxygen, okay? In this diagram, they have then the rings that are around the vascular tissue. This is where then the carbon, the C4 organic acid, is moved into these bundle sheath cells where it's decarboxylated, and then the normal C3 metabolism occurs in these cells. And the purpose of that is to raise the CO2 to eliminate the inhibition by the oxygen, or photorespiration, okay? And then, okay, I wanted to go through this one just real quickly. So in C4 then, again you have these cells here that have a different kind of chloroplast than you're going to find out here. And so here we have the mesophyll cell where we have the actual first carboxylation of PEP with CO2 to produce this 4-carbon compound, which is then moved into the bundle sheath cell where decarboxylation occurs. The CO2 enters then the normal PCR cycle, okay? So in the mesophyll cells, there's not any rubisco. In the bundle sheath cells, that's where you're going to find rubisco and where the normal PCR cycle is occurring, normal. The C3 cycle is occurring. Does that make sense? So in the case of C4s, you have two kinds of cells, two kinds of chloroplast in the cells. Okay? And I wanted to go down to the end of this just to once again give you a comparison that in the CAM plants, the C4 metabolism occurs at night, so CO2 comes in at night, and organic acids are stored in the vacuole. Then during the day, the stomates are closed. The stomates are open at night, obviously, when it's cooler. And then the C4 organic acid is decarboxylated and moves into the normal C3, the C3 cycle. I keep calling it normal, okay? And I wanted to just finish up with this part. CAM plants have a much lower transpiration rate, obviously. They're in the desert, and a lot of CAM plants really don't have normal leaves. They've got some kind of a modification. They've got really small leaves, or the leaves have become part of the plant body. And another important point is carbon assimilation is much lower in CAM plants than in most C3s and obviously all C4s. Desert plants don't, in general, grow very fast. The exceptions are, and I couldn't find, there are these seeded plants that when there is an occasional rain, they germinate incredibly rapidly. They grow extremely fast. And I tried to find the name. I can't find the name of a single one of them. And produce seeds in a very short period of time. So basically, you know, the function of a plant, if you go back to what E.O. Wilson says, our function is to produce offspring. It is to produce the next generation. So these plants, these very few plants have adopted this incredibly rapid ability to germinate, grow, produce seed, and produce the next generation. Okay? Questions about that before we start talking about how carbon is moved around the plant? So today we're going to do translocation. Then Wednesday we'll be talking about metabolism. And that's going to lead into secondary metabolism, which is a chapter in the end of the book, but I like to talk about secondary metabolism of metabolism. And then we'll go from there, okay? So now we're going to talk about what happens once the carbon is fixed. And what you're going to see is that a lot of carbon is moved around as C3 organic acids, or as C3s, triose phosphates. And that if we need to move carbon out of the chloroplast, then that triose phosphate is converted into glucose, and then in most cases the glucose is converted into what for transport? What's actually transported in most plants? From the leaves through the stems down into the base of the plant. What carbohydrate are we talking about?
Glucose.
Not glucose. Sucrose, okay? It's 12 carbons, and the big thing, and I've mentioned this a couple of times, glucose and fructose, if you put them together, you make a sucrose molecule. And that's what, remember, invertase will break down. A glucose molecule and a fructose molecule are very reactive. So you're not going to find glucose in the cell, you're not going to find fructose in the cell. You're going to find glucose 1 or 6-phosphate or fructose 1 or 6-phosphate. With sucrose, those reactive ends are stuck together. So sucrose is a fairly stable molecule. And you're also moving 12 carbons every time you move a sucrose molecule. Okay? When you go to the store and you buy a 5-pound bag of sugar, what kind of sugar is that? Sucrose. Why don't you buy fructose or glucose? Why are you buying a 5-pound bag of sucrose? Why sucrose? What do you think? Come on. Stability, but why? Why are you buying it? Huh? It's cheap. You know, it's cheap. Plants don't store fructose or glucose. Most plants store sucrose, and the two big plants that we use for sugar are sugar beets and sugar cane, which both have a higher concentration of sucrose than most plants, obviously. If they didn't have a higher concentration, we'd be using another plant. Okay? The only time you actually get fructose or glucose, and it's almost always fructose. Fructose, when you taste fructose, fructose tastes sweeter than sucrose. So if you use any kind of artificial sweetener, the 99.9% of that packet when you open it up is fructose. The actual chemical that's the artificial sweetener is very few molecules compared to that packet. You know, in fact, you couldn't have a packet of most artificial sweeteners without some kind of carrier. So the fructose, the white powder you see in there is fructose with the chemical sort of laced in there. The only one that's you actually have a 1-to-1 ratio is Splenda, which is a modification of sucrose. So it's sucralose. It's sucrose that's been modified. Okay? It's a little sweeter too. So what we're going to talk about now in more detail, and I told you would be keep going over these over again, is more detail about phloem and actually how the complex pattern of carbohydrate movement, and then probably we'll finish up, I don't know, we should get to it today, is a little bit that's, it's not new, but it's become newer research, and it's xenobiotic compounds. And basically what we're talking about there is usually herbicides that can be moved through the phloem. So we're going to first start with the two things that you know most about, which is starch and sucrose. And starch is how carbon is stored for a long term. And starch, if you remember back, one of the first lectures we talked about starch and cellulose. Cellulose is beta 1,4 glucose molecules stuck together. Starch is basically alpha 1,4 glucose molecules in another form. But starch is made in the chloroplast, and then if the carbon needs to be moved out, then triose phosphate can be converted either into hexoses and sucroses, or triose can be moved out. Okay? Now, a term you will hear a lot is carbon allocation. And that term refers to what's going on in the chloroplast. The first thing the chloroplast is going to do is keep that cycle going. So remember, once CO2 is fixed, that entire cycle has to go. So over time, when there's a lot of sunlight, then you're going to build up of sugars, and that excess sugar is going to move out of the chloroplast, and that carbon is going to be allocated somewhere else. It's going to be moved down into the roots. It's going to go to the growing tips, wherever energy is needed. Okay? Now, again, starch is the form that is stored, and we have actually two kinds of starch. We have amylose, which is the alpha 1,4 links. And then we have another kind of starch that's very common called amylopectin, which has an alpha 1,6 branch. And starch basically is a mixture of these two things. And different starches from different species will have different combinations of amylose and amylopectin. And if you can think of what starch is, it's really compact. So think of like starch being beads, you know, and each bead being a glucose unit. And you're just basically wrapping those things together, and they're incredibly dense. So you're able to store, the plant can store a lot of carbon in starch. It's also not reactive. And so it's a massive amount of carbon as that starch molecule. Now, how do things move around? The most common way that carbon is moved around from the chloroplast to the cytoplasm is by a triose phosphate translocator. Okay, guess what? It's an integral membrane protein that takes a triose phosphate, which is, if you remember, that's the first stable product of the dark reactions. And as excess triose phosphate is formed, it is moved out of the chloroplast. We're also going to see, either in this lecture or another one, that glucose can be moved out. So it's a translocator. Triose phosphate moves out, okay? And if you can see here, it's a little small. A phosphate moves in. Why would you need that? You have a triose phosphate moving out, and you have a phosphate moved in. So it's an antiport membrane protein. Why is that important? Huh? Why is that important? Why does that make sense? Maybe that's a better way of asking the question. Triose phosphate moves out, a phosphate moves in. Right, so you balance in the phosphate. If the triose phosphate just moved out and phosphate didn't move in by this mechanism or by another mechanism, then very quickly the chloroplast wouldn't have enough phosphate to work with. So the beauty is you're actually moving 3 carbons, and that's it. And then this enters the pool, the hexose pool, and we're going to be talking about this today, and we're also going to be talking about this, we're talking about metabolism. And if we want to move it from this cell, then that hexose is converted into sucrose, which is moved long distances, okay? So sucrose is made for transport out of the cell containing the chloroplast. Okay? And if this were a C4, this would be, the phloem would be right here, and that sucrose could very easily be loaded into the phloem. Remember, C4s, it's not a matter of just photosynthetic efficiency. It's the fact that the transport system is right next to the cells that produce the sucrose, the rings of chloroplast right around the phloem, the vascular tissue. So we've got a lot of things that make C4 plants more efficient. Now, how do we know where these things are produced and moved? Well, you know, if you do this to a plant, if you do this to a tree where you are cutting through the bark, which is the phloem, and then it's separated from the wood, the xylem, by a cambium. What happens if you actually girdle the tree? What happens? Why does the tree die? Huh? Oh, water. You still got water transport through the xylem. What's happening? Where does the tree die first? Where? But where? If this is the bottom of the tree, this is the top. Where are the cells going to die first? Right. The base where the sucrose is being moved to, you no longer have sucrose being moved down into the roots. So you have a deficiency in energy. Now, there are going to be a lot of other reasons it dies, but the main thing is that you're cutting off the transport of sucrose into the plant. Now, if you like maple syrup, you know that they'll make a slight V in here, and they can collect the actual sap from the phloem, which contains the sugars. Not very much sugar. I was listening to a podcast the other night. I think it takes 20 gallons of sap to make 1 gallon of maple syrup.
Yeah, we have a sugar shack in Michigan.
Oh, really? Okay.
Yeah, and just depending, like sugar maples, which have the most, it's about like 20, 21, but like most of your maples are going to be like silver or, a variety of them, and it's going to be like 5.
5 to 1, okay.
Yeah, or like 51, or like it takes a lot, so.
There's a new method, by the way, that they've just introduced where, because you've got to wait for the tree to grow, become mature, before you can harvest the sugar, the sucrose solution. This new method is you just use saplings, and you grow them, then you plant them as a crop. You cut the tops off, you put a vacuum device over the top of the sapling, and you pull then all the sugar out. And you get a much higher yield. You don't get as high grade of maple syrup as you would with a normal mature tree, but you can produce a lot more quickly. Now, another thing that was done pretty early on, and I think we can flip back here, is we can add C14. And here is a picture of what one would see if a stem was cut or petiole was cut in cross section, and you would put this on a photo emulsion, and this is C14 loading, and of course you're going to see the dark. So you add radioactive CO2 to the leaf, cut the stem, and where does the dark show up? It's going to show up in the phloem. So this shows you that the C14 that's fixed from the atmosphere ends up as sugar, and then it ends up in this transport tissue. So we're pretty sure, we know, that sugars and a lot of other things are moved through the phloem. Remember, the big difference between phloem and phloem is living tissue. Very complex. We're going to go over even more complexity of phloem today or Wednesday. Remember, so phloem is living tissue, and that's where sugars are moved. This is what we find. Sucrose, other sugars. There are proteins, there are amino acids, there are organic acids, there are some minerals, ions and cations. And also, as you're going to see in the later part of the semester, that phloem is also the location of a lot of movement of phytohormones, because since it's living tissue and you can have proteins in the membranes, you can move phytohormones up or down. You can move them around. Now, just to give you just some idea that there are some plants that produce and transport other things. Some grasses produce raffinose, which is just another complex carbohydrate. And then some plants, like cherries, produce mannitol and sorbitol, which you may have heard of. And the sorbitol is used a lot. Sorbitol does not have a high. We don't use sorbitol very efficiently, so it's used in a lot of sugars, candies, and stuff that are, quote, sugar-free. So the sorbitol has some sweetness, the mannitol has some sweetness to it, and so it's used a lot in diabetic foods. So you'll see something that says sugar-free, not a low-calorie food. Okay, it plays better with the body than other sugars. Now, if we look at what we can find in the South, we have most plants have this, which is sucrose. Some plants have raffinose. Some plants have stachyose. And then some plants have verbascose. Don't worry about these. You find these in cucumbers, or you used to find these a lot in cucumbers, these longer carbohydrates, which is fructose, glucose, then a galactose, then a galactose, and then a galactose. We don't digest these well. So what happens when you don't digest something well? You burp. So you see burpless cucumbers, which you still see some now, but in general, over time, these more complex carbohydrates with these galactose units, they've lowered the concentration of these so that you don't have that problem. You don't belch or burp. Now, let's look at more detail of what the phloem actually looks like. Oh, I didn't realize that was covering that up. Okay. You've seen this before. Okay. You have the sieve elements and then the tube members. So you have this living tissue. And what's unique about this larger, the sieve tube? What's unique about this living tissue? What does it not have? Anybody remember? This was on exam 1. Does not have a nucleus. So in addition to this, we have to have a companion cell. And the companion cell has a nucleus, and it's where a lot of things are made, and then those products are moved in for transport. So this larger member here is where the stuff is actually transported. And then a third member we'll have is called a phloem parenchyma cell, which is just a large cell that supplies material to the phloem. So a little more complex. Remember, these are cells, so when we're moving across here, there might be some openings, but basically these cells are moving from membrane to membrane. So to move from here to here, in most cases, requires whatever's being moved to be moved with a protein, a transport protein. So what you're going to see is like some hormones will move here, and then they'll move down here, and they'll actually be carried down when we talk about hormones, okay? Okay, so that's a little more structure than you had in the past, a little more complex. And let's talk about some terms now, sources and sinks. A source is very simple. Where's the carbohydrate coming from? It could be a leaf that's making sugars, or it could also be the breakdown of starch, let's say stored in a tuber, to move into the plant, provide nutrition or provide energy for the plant. Remember, the carbohydrate in plants providing energy, providing backbones for all the other molecules in the plant. And they can change. A sink is where the carbon is moving to. So the sources and sinks will change over time. A lot of people that are basically modelers look at source-sink relationships because that's very important to yield, to crop growth. So a lot of modelers will, take all these reactions and they can determine at what point when a leaf is very, very small, it's a sink. It's not making enough carbon to grow. So it's transporting sucrose into the small leaf, then the leaf grows, and then the leaf starts transporting carbon out. It becomes a source, and then finally, of course, then the leaf is mature, photosynthesis goes down, and eventually the leaf dies. So they change all the time. Now, let's talk about one of the mechanisms that can be used to explain how carbon moves, how sucrose moves. And it has a combination of xylem and phloem involved, and has a combination of pressure and water movement. So guess what? We're back to water relations. What moves one way in the xylem is water. And of course some minerals, a lot of minerals of course. So what seems to happen then is you have this water here at a fairly high concentration under pressure. And you have sucrose that's produced. So as sucrose is produced, in this case this cartoon is a picture of this is supposed to be a leaf, and the sucrose is moved into the phloem. What does that do to the water potential of that part of the phloem? We just had it. Does it go up or down? Does it become more positive or more negative? It becomes more negative, which then means that water can flow from the xylem, which is, remember, they're not separated by space. They're next door to each other. They've of course got a separation layer. So the water moves in here, which then tends to cause the flow of the flow to move down then, okay? Then the sucrose is removed at the sink. Let's say this is a root or tuber, which then causes the water potential to be lower, and boom, and it moves some. So we have sort of a bit of a cycle here. Of course, we've got a lot of water moving up here at the same time, okay? So water, following the water potential gradient, diffuses into the CO2 members from the xylem, okay? So it's very, very simple system. Well, I won't say simple, but involves it. Now, we're going to go with the same system again. This is called the pressure flow, and it has a lot to do with not just movement of the water, but the production of the source and then the removal of the carbon in the sink, okay? So you continuously then have a higher concentration of sucrose from the source into the xylem, into the phloem, and then the movement from the other end, okay? And again, then that's going to cause the water to move back through. And this is called the pressure flow hypothesis. Okay, questions? Questions? So to go over it again, in this cartoon, this is the source leaf. So you're going to have a high concentration of sugar, moves into the phloem, which then is going to cause water to move from a concentration of more water into lower, so from a higher water potential to a lower water potential, and then this pressure then moves the flow down. Okay? Then at the other end, the water can be moved back into the xylem and move up. So remember, xylem basically is one-way transport, but phloem, we can have, in this case, sugar is moved down the phloem, but sugar can also move up in the phloem. Things can move up and down. So the phloem is bidirectional. The xylem is, again, from the bottom to the top. And we can actually make a system, okay? So, and we've done this. We used to do this in the lab when we had a plant phys lab, that you can actually show that it works. And you can take two flasks and connect them so that there's no air spaces. So this is a continuous stream of water. Remember, if we had an air space in here, nothing would move. And then we can put dialysis tubing, and we can put in this dialysis tubing sucrose. Okay, what's that going to do to the water potential inside the tubing? Become more negative. We're lowering the concentration of water. So if it's pure water here, which way is the water going to move? It's going to move in. And in this system, that's going to produce a pressure so that water is going to flow. And you will actually see, and what you have in here is sucrose in a dye. And of course, the blue here is really clear because it's water. And you can actually see the dye start moving through the tubing into the other side. Okay, so you can make one of these things, and they work pretty well. Does that make sense? You just have dialysis tubing with the sucrose and dye in here, and the blue is, of course, clear because it's water. And you set this thing up. You have to be very careful that you don't have a single air bubble because, of course, an air bubble means that nothing's going to move. Remember, cavitation. So you set it up so that you have no air bubbles, and you can actually watch the dye slowly move to this tube, to this, until this side. Now you won't go continuously because you don't have a new source of sucrose, but it makes a really nice lab demo, okay? Now do you understand how you can follow that? Again, we're back to water potential. Water's going to go from higher to lower concentration, and when it moves in here, it's going to generate some pressure, which is going to move it up into this tubing. And you literally can watch the blue, usually, I don't know why we use blue, but you can actually see the blue moving up in the tube. Okay? And you can do this in an afternoon plant phys lab. You can set it up, go away, study, come back, and you can actually measure how fast the dye is moving in the tube. Okay.
Now that changing water potential, does that do with the specific gravities of the solution or the compound, or is it just?
No, it's just basically due to the sucrose.
Oh, okay.
You put a tremendous amount of sucrose in here. So this is like something you'd never find. This is like syrup.
Okay.
So you're forcing the experiment. So you put enough sucrose in here, well, I'll say syrup. It's maybe 10%, 20% sucrose. So it's maybe 10 times more than you'd have a glass of tea. And so you're forcing, so the water potential between here and here is large. This is zero or near zero since it's pure water, and this is minus, fairly minus. So you force the experiment to go. Of course, the larger the difference between the sucrose and the water, the faster it's going to move in. Okay? And you've got to be sure, and out of 4, that 5, 1 or 2 will fail because there'll be a rip in the membrane. But usually out of a lab, you can get 3 out of 4 to work fairly easily, Okay, now, again, the movement of the phloem is bidirectional. That's going to become very, very important when we start talking about hormones. And now we're going to talk about, we'll talk about allocation and partitioning next time. I want to stop here before we get to that. So let me back up here. We're talking about sources and sinks, and then we went over how that was done. And now what we're going to finish up with on Wednesday is how the carbon, the sucrose, the stored carbon is actually where it moves into the different parts of the plants, where it's allocated, and then where it's partitioned. And these are terms that people that look at the whole plant level of things, like a lot of bottlers, really look at. Because we want to increase, let's say, the sink strength of a developing seed. So maybe we'll get a larger seed that may have more, not just carbohydrate, but more everything in it, okay? So I will see you next time then.